In this video, I'm going to be talking to you about Data2 background. So Data2 stands for Divisive Implicance Denoising Algorithm, is an open source algorithm implemented in R which uses a statistical inference to correct applicant errors. It attempts to simplify the study of microbial communities by allowing to reconstruct applicant sequenced communities as at the highest resolution. Data2 implements a complete workflow that takes raw applicant sequencing data in FASTQ files as inputs. It produces an error-corrected table of the abundance of applicant sequence variants in each sample that we call ASV table, which stands for Applicant Sequence Variant Table. So Data2 has some benefits. It's compatible with all applicant types, 16S data, but you could use it on 18S data, ITS, which is internal transcribed spacer, with some slight modifications to the pipeline. Even though in the beginning Data2 was designed to work on Illumina data, you can actually use it also on IronTorrent and Roche 454 pyrosequencing as well. Data2 gives single nucleotide resolution of applicants that allow the better identification of chimeric sequence. It takes into account the error rates and it lowers the amount of OTUs, which will lower your false positive rates. So Data2 is implemented on R and RStudio. It is also integrated, simplified, and condensed in QIIME2. So most of the steps of Data2 workflow are condensed into more or less 2 lines of code in QIIME2. If you are new to the field, QIIME2 could be a good option to start with because it gives a graphical user interface, and then you can use Data2 workflow in R and be able to customize it according to your data and your analysis. So this slide summarizes the theory of microbial community analysis. So in a particular environment, let's say we have 4 specific number of samples. So in this figure, we have 4 unique sequences. So during the step of amplification, some errors can be introduced, which creates some similar sequences, but they are slightly different, as you can see here. So those sequences introduce some noise in your data. So with the OTU picking, so OTU stands for Operational Taxonomic Units. So it's cluster the sequence according to their similarities. It's usually at 97% threshold, and you can modify it to 99% or 94%, and also according to which OTU picking strategy are you using. I won't go into too much details. So the output will be different clusters of OTUs that group similar sequence. So in this figure, so we have 2 OTUs as a result. So even though the red and the green sequences are from different organisms, as you can see here, the OTU picking will put them in one OTU cluster. And the same case for the blue and the gray sequences. So Data2 pipeline is trying to detect this error before doing the picking or clustering of your sequences. So the main step of Data2 is sample inference, which based on your sequence and the quality score and their abundance, it makes a call as to whether or not your sequences are real or not. And based on these steps, Data2 resolves some of these errors. So let's move to the next video.